Timothy Karr from Kyoto Genetics Talk Center.
Thank you very much. Well, I know what it's like to be the token male in a session. Oocyte, oocyte, oocyte. And I was wondering if I can attest to the long flight from Osaka to Orlando. I was wondering if I could get a 2-minute credit for jet lag and, no? Okay. Well, anyway, get organized here. It's a pleasure to be here, and I'm looking forward to talking about sperm and spermatogenesis to you today. Let's see here. And I've got to start off, this being the TAGC, Totally Awesome Genetics Conference. I'll start off by actually talking about, or using a word that doesn't belong in Drosophila lexicon yet, and that's the epididymis. We'll be talking about that at some length today. And I want to first start off, though, by acknowledging my colleagues who have helped enormously in this project. Oops, what did I do? I didn't do anything.
I'm not sure you did.
I hope I didn't. Let's try that again. Yeah. So the transcriptomics and RNA profiling work was done by colleagues Brian Oliver and Leif Benner at the NIH. And the asterisk here by their name means that they're here at the conference. So please, any questions about this sort of high-end RNA-seq profiling, you're more than welcome to direct them to Leif and Brian. And on the proteomics side, a former PhD student, Sherry Skirgit, and Matt Roseno, who's a proteomics expert, have done that. And together we've surrounded this structure, which I'll be talking about today, which is, hopefully we'll be able to convince you, is the equivalent, or possibly the equivalent, in part or in full, to an epididymis, or an insect epididymis. So what is an epididymis? We have to start off by first defining that. If you don't know, it's a structure that all mammals have, males, that lies along the outside border of the testis. And all sperm that are made in the testis exit the testis and must travel through this tortuous long tube located inside this epididymis. And there's basically 3 major landmark features of an epididymis called the caput, corpus, and cotta. And this is actually quite an ordeal. This tube can be up to 2 meters long in humans, for example, and it can take many days for sperm to traverse this gauntlet, if you will. And why is there an epididymis? Why make a bunch of sperm on the one hand and then just put them right back into a sort of a torture chamber that they must travel through? And the reason is that sperm that come out of the testis, although they look like a sperm, they swim like a sperm, they're completely incapable of fertilizing an egg. And they must traverse through this pathway down to the cotta to acquire fertilization competency. Presumably, this process results in some kind of modification of sperm. And work from Sherry, her PhD thesis, was actually looking at the changes in sperm proteomes that occur during transit through the epididymis in both the mouse and the macaque. The mouse, this is published, and the macaque sperm proteome is published, and she's working on this same kind of study in the macaque. And what's surprising about this is just how massively these sperm are modified. By just doing accounting, protein accounting of the 3 proteomes, there's about 1,000 proteins are swapped in and out of sperm during this transit. And the same thing is true in the macaque. The other feature that's common between the two is that the mature sperm that are fertilization competent contain far fewer proteins than the sperm that come out of the testis. So there's some kind of pruning process that's going on, and clearly what goes in is not what comes out. And if you want to learn more about this, we have a poster up for this meeting. But how do we get here from the epididymis? How does this structure relate to what I just told you? And it all started, this story all started with a proteomics project, which we've been working on for a number of years in defining the Drosophila melanogaster and other Drosophila species proteomes. And what became very clear very early on was that if we just looked at the expression of genes in the testis that encode the sperm proteome, more than half of them are not expressed highly in the testis. The ones that are overexpressed you'd expect is a very low number, and it raised the question is where are these proteins coming from if they're not coming from the testis proper, and led us to start thinking about other structures, and of course that would be the seminal vesicle, and which I'll talk about as PE-SV region. And of course this idea is exactly what's happening in the epididymis in mammals. Proteins are being inserted and taken out of the sperm in massive quantities in mammalian sperm maturation. So, but there's a big problem with this analysis and this conclusion is because it's really based on fly atlas data, which doesn't include a seminal vesicle. So it was basically an indirect inference that these proteins that are underexpressed are somehow expressed somewhere else. We did not know where. So it did get us thinking about the seminal vesicle, though, as a possible source of this. So I thought I'd just go to the literature and read up on the seminal vesicle, but there's just no respect and no love for the seminal vesicle. If you just look at the number of citations in the last 5 years, 1% of them have the word seminal vesicle associated with them. Everybody's been, for obviously good reasons, focused on the accessory gland in the testis. And, but I did find one beautiful paper done by Tokiyasu and Hardy back in 1972, where they looked at this terminal region here, which they call the terminal epithelium, which is where sperm attach to this region and coiling of the sperm occurs and individualization begins. And so that gave us something to think about. And as I'll show you, as this picture shows you, this terminal epithelial region is probably a little larger than this diagram drawn 40 years ago, and may include a slightly larger region. And so we had good reason to think about this region as being the source of these proteins. And so to begin a study of this and to see if there's any relationship, a correlation with the mammalian epididymis, we took and did by standard LC-MS proteomics, just determine what is in this tissue region, and then just using this list of proteins and looking at a distribution of the kinds of GO categories that are enriched in these, it became very clear that it looked very much like an epididymis. It's dominated, unlike the sperm proteome, which is dominated mostly by metabolic, energetic, and cytoskeletal elements. This is dominated by plasma membrane systems, apical localization, endoplasmic reticulum, coated vesicle, Golgi protein targeting systems. So it's starting to walk and talk a little bit like a duck. But we still needed direct evidence that the underexpressed genes encoding the DMSP proteins are expressed in this region. And that's where we called in, fortunately, my colleagues, and I was able to go visit the NIH, and with Leif and I were able to dissect this region specifically out of the testis, seminal vesicle, and TE region, separate them, and then Leif was able to do his magic and generate a very nice RNA-seq library, and the results are quite clear, actually a little more dramatic if you just put a dark background here for people like me who are partially colorblind. it's quite clear that there's a very distinct difference in the expression of genes in these two tissue types. And in fact, we could then look at the sperm proteome taken out of this and see that there's a very large proportion of proteins that are in the sperm proteome that are actually expressed outside of the testis proper. And by looking at a difference map of those proteins, what are they? How are they different? They're all in the sperm proteome, but how are they different? We again just used Cytoscape, a common program for visualizing these GO categories and enrichment of them. And it's quite clear that if we just looked at just a difference map, where the program simply compares the categories in each of the two groups and gives you a heat map of whether they're enriched in one or the other, it was quite clear that they fell into two very distinct categories. Some of which I've indicated here, that things that are upregulated in the SETV are things related to polarized membrane components, like I've already shown, endocellular plasma membranes, et cetera, and likewise in the testis, things related to mitochondria and energetics. And you see most of this is gray, which means they're about equal between the two. And this particular group was quite illuminating with respect to thinking about the functional differences between these two regions of the sperm biology. And that is that this group of comparison between the upregulated in the testis and seminal vesicle actually fell into two very distinct categories related to the ciliary part. They're all related to aspects of cilia, of flagellum, of flagellar components. And one is clearly related to testes expression, and one related to TESV, this terminal epithelium seminal vesicle region. And one can think of this, or begin to think of this, as the testis proper making the building blocks for a sperm. And these specializations, which can be thought of as specializations, because they're really specialized proteins that were first identified in neurons and neuronal biology. And of course, there's a central region of proteins that are common to both of them. And so this was a very nice result, because our work in the mammalian epididymis has a very similar kind of patterning. As immature sperm become mature, they acquire these specialized components. So it's beginning to walk like a duck, talk like a duck, and I'm almost out of time. I am the last speaker on. Okay, I'll hurry up, but I'm out of time. So we decided to look at some of the CPTI lines that overlapped with the Cambridge protein trap insertion lines that allow us to look at GFP expression where the protein is expressed. And this is just a picture of a common protein that we know is expressed exclusively in the testis, DON1, and in the testis and the seminal vesicle, you see very high levels of expression in the sperm tail, which you'd expect. But just some of the proteins we would predict would be expressed in this TE transition zone clearly are expressed right where we'd predict they are. This is one is a poly-A ribosomal binding protein, or RNA binding protein. And glutamate dehydrogenase, surprisingly, is expressed highly in this region and has some very unusual structures that we've never seen before, but are indicative of the kinds of processes that might go on in a specialized region like an epididymis. And finally, there's a regulator of receptor-mediated endocytosis that actually is expressed along the duct where sperm exit out of the testes into the seminal vesicle. So it's really, we have a working hypothesis that you have a caput corpus incauda and an epididymis, and in this region of the terminal epithelium or transition zone and the seminal vesicle, you have, and when you actually dissect and look at these structures carefully, you start to see some substructure in this region at the end of the testis. And it really kind of breaks down into three sort of morphological features. Now, I'm not about to call it a capita, a corpus, and a cauda yet, because we haven't yet established that precisely similar processes are occurring. So, but I've got to call them something, so I thought I'd call them Mary, Low, and Mo, Larry, and Curly for the purposes of today's talk. You can see a nice gradient of expression along this structure of this protein. And so in summary, our proteomics analysis that started off as a simple sperm proteome has led us to start thinking about this region as being the equivalent of an insect epididymis. And from Japan, there's 12 different ways to say thank you. And thanks for your patience and listening to this talk.
Questions?
So when you dissect these out and you do the transcriptomics or the proteomics, it seems like you're going to have the tube structure that these sperm are moving through as well as the sperm. So the question is there a way to distinguish the differences in the proteins that are autonomous to the sperm versus the vessel that they're traveling through?
Yeah, the sperm proteome that we defined was actually from sperm, pure sperm taken from the seminal vesicle. So we know what that is, and we can go in when we do our proteomics of that region, we can subtract out the sperm proteins and look at what's left, and that's what we did. Yeah.
That's one.
Do you observe a hyperactivation of sperm as they pass through this region of the testis?
So this is one of the interesting things about Drosophila sperm biology and fertilization biology in Drosophila is that we really, you can't do those kinds of experiments in Drosophila. We can't easily obtain pure sperm and manipulate them the way that you do with mammalian sperm because they're 2 millimeters long, and it's like trying to manipulate spaghetti. And so looking at those kinds of things are difficult. In fact, we don't even know that sperm that come out of the testis, that at that point, are they fertilization competent? Must they go through? And that's what we're working on now, trying to figure out if we can identify proteins that must be inserted into the sperm as they exit the testis and into the seminal vesicle to answer questions like that. Well, no one's done that in Drosophila or any insect that I'm aware of where actually we're able to show differences in fertilization competency because basically you make sperm in a testis and it just goes into a seminal vesicle. And the idea of an epididymis or a structure post spermatogenesis that's actually modifying sperm is what is new here.
Okay, I think there are no more questions. Thank you, Tim, for this wonderful presentation. We'll like to thank all the speakers of today's session for their wonderful presentation, and this is the end of our session. Thank you all.